Hello, everyone. Food is the paramount necessity of the people, and food production begins with seeds. Seeds are the foundation of agricultural development. Some people refer to it as the chip of agriculture. Today, I would like to share with you the related content of seeds. We will cover three aspects: the structure of seeds, the main types of seeds, and seed germination. Let's start with the structure of the seed. The seeds of different plants differ in shape, size, and color, but their basic structure is the same. The seed is generally composed of three parts: seed coat, embryo, and endosperm. Some seeds have only the seed coat and the embryo. The embryo consists of the plumule, hypocotyl, radicle, and cotyledon. The seed coat is the protective layer on the outside of the seed. Mature seeds often have a hilum and a micropyle on the seed coat. The hilum is the scar left where the seed detached from the funiculus. At one end of the hilum is a small opening called the micropyle. Some seeds also have structures like the raphe and carbuncle, such as castor bean seeds. The embryo is the most important part of the plant seed. The plumule is located at the apex of the embryo and is the primordial structure of the future shoot system, which will develop into the above-ground part of the plant. The hypocotyl is located between the plumule and the radicle, and is connected to the cotyledon. It later forms the connecting region between the root and the stem. The part from the cotyledon to the first true leaf is the epicotyl. The part from the cotyledon to the root is the hypocotyl. During seed germination, the elongation of the hypocotyl helps the cotyledons of some seeds to emerge from the soil. The radicle, located below the hypocotyl, is conical and represents the embryonic primary root within the seed, which will later develop into the taproot of the plant, and is the first to emerge from the seed coat and form the root system of the plant. The cotyledon is the embryonic leaf or seed leaf, usually one or two, attached to the hypocotyl. Based on the number of cotyledons, plants are classified as monocots, dicots, and polycots. The endosperm functions to store nutrients that are digested, absorbed, and utilized by the embryo during germination. The endosperm of some plants has been absorbed and utilized by the embryo during development, leaving no residual endosperm in the mature seed. The main nutrients are starch, fat, protein, and so on. Starch seeds such as rice, wheat, corn, etc. Oil-rich seeds such as peanuts, rapeseed, sesame, etc. Protein seeds such as soybeans. Based on the presence or absence of endosperm, seeds are classified into two types: endospermic seeds and non-endospermic seeds. It is further divided into four categories according to the number of cotyledon. This is a castor bean seed, an example of a dicotyledonous endospermic seed. Castor seeds can be pressed for oil. The oil content is about 50%. Castor oil is an important industrial oil, can produce surfactants, is also a high-grade lubricating oil raw material. The outermost layer of the seed is the protective seed coat. Inside are the embryo and endosperm. The embryo consists of the plumule, hypocotyl, radicle, and cotyledons. This is corn, known as a longevity food, is an important food crop and feed crop, and is also one of the highest yielding crops worldwide, with planting area and total yield second only to rice and wheat. The corn seed is actually a fruit, a caryopsis. Its outermost layer is the pericarp fused with the seed coat, which are tightly fused and difficult to separate, and are conventionally referred to as the seed coat. Therefore, more accurately, this is the fruit of corn. Inside the seed coat is the endosperm. The starchy endosperm, along with the protein-rich alurone layer, constitutes about 80 to 85% of the seed's weight. It plays the role of storing nutrients and supplying seeds for germination. The embryo consists of the radicle, plumule, hypocotyl, and a single cotyledon. This is a broad bean seed, an example of a dicotyledonous non-endospermic seed. The outermost layer of the seed is the seed coat. Inside is the embryo, comprising the plumule, hypocotyl, radicle, and cotyledons. The cotyledon is the place where nutrients are stored, which is the part we commonly refer to as the bean in broad beans. Seed germination refers to the process by which a viable seed's embryo from a dormant state to an active state under suitable conditions and begins to germinate and grow to form a seedling. The process of seed germination involves three stages: 1. Imbibition, i.e., water uptake and swelling; 2. Radical emergence, i.e., the radicle breaks through the seed coat; 3. Seedling establishment, where the radicle length equals the seed length and the plumule length equals half the seed length. Generally, the radicle first breaks through the seed coat to form the taproot, followed by the plumule breaking through to form the stem and leaves together with the hypocotyl, ultimately developing into the whole plant. Seedlings can be categorized into two types based on hypocotyl elongation, according to different growth rates of cotyledon unearthed seedlings: epigeal germination and hypogeal germination. Cotyledon unearthed seedlings, characterized by hypocotyl elongation, pushing the cotyledons and plumule above the soil surface. The picture shows a bean seedling with epigeal germination. Other representative plants include soybeans, cotton, rapeseed, and cucurbits. Hypogeal germination, characterized by the hypocotyl not elongating, the cotyledon remains in the soil. Epicotyl growth pushes the plumule above the soil surface. The picture shows a pea seedling with hypogeal germination. Representative plants also broad bean, lychee, wheat, corn, and so on. Knowing the type of plant seedlings has important guiding significance for agricultural production and can be used as the basis for correct planting depth of plant seeds. Under normal circumstances, seeds with hypogeal germination can generally be sown slightly deeper, while seeds with epigeal germination should be sown shallower. Of course, seed size, soil moisture, and other conditions must also be considered, comprehensive factors to determine the actual depth of sowing in order to improve the emergence rate. Let's summarize the similarities and differences between dicotyledonous seeds and monocotyledonous seeds. The main differences are the presence or absence of endosperm and the storage of nutrients are different. The similarity is that both have a seed coat and an embryo. Now, let's do a little test. You should have heard a riddle about peanuts. It goes like this. Hemp house, red curtains, there lives a fat white man. So, let's see what part of the peanut we're talking about. First, the hemp house is the outermost structure of the peanut fruit, that is, the peel. Then the red curtain, a thin layer of red inside the peel, is the seed coat. The white fat man at the bottom is actually the embryo of a peanut. Finally, to borrow a phrase often used by Professor Zhong Yang, that is, a gene can save a country, and a seed can benefit thousands of people. I hope you can be a solid and full seed to spread to the people. That's all for today's lesson, and I'll see you next time.